draw it, just go back to the picture, is that they can start translation before transcription is ever finished. Right, that's the cool thing about prokaryotes. So as soon as any mRNA is made, they can start their transcription or translation. Why can't eukaryotes do that? There's a bunch of reasons. Why does translation have to fully finish, or why does transcription have to fully finish before translation begins in a eukaryote? Good marker. There's your eukaryotic cell. Where's transcription happen? In the nucleus. Right? So there's your DNA.
So that's why, right?
Right?
It's not happening in the same place.
Right? There's your beginning of your RNA. And your RNA has to leave the nucleus for translation to occur. Here's our protein. So you can't have coupled transcription translation in a prokaryote or in a eukaryote. So for all of these, look for the similarities, look for the differences. Okay, what do they have that's similar? What do they have that's different? Where we finished, or we were just beginning to talk about, is genetic regulation in prokaryotes. Prokaryotes, almost everything is regulated by making RNA or not making RNA. That's what we say at the level of transcription. Remember prokaryotes? They had this thing called an operon. Operon. 3', 5'. All right. I'm going to put my genes right here. And an operon can have multiple genes. So we're just going to have A, B, C, and D because we always, for some reason, letter them. Where does RNA polymerase bind that drives transcription? What do we call that site?
It starts with a P.
It does. Right here, starts with a P.
The promoter.
A promoter. So this is where our RNA pol binds.
Not looking.
Where does transcription stop? What do we call that site?
The terminator.
The terminator. There's our terminator. Okay. What we started talking about at the end of class last week was how this is regulated. We had our constitutive, where they are always on. We have our inducible, where they're usually off, and then we can turn it on. Or we have our repressible, where they're normally on, and you can turn it off. The way this is turned on and off is by repressor proteins. And so if you remember, upstream here, we had a gene labeled I, and I encodes the repressor protein. Whoops. And let's make the repressor protein a triangle here. Right? So your repressor protein is this triangle. Let's just say this is an inducible operon. Something that's normally off and we're going to turn on. If this is turned off, where would our repressor protein bind? That was kind of where we ended last week.
Promoter.
It doesn't bind, it's not the promoter specifically. It's something just downstream of it. It's a site here called the operator. So what happens, with an inducible operon, and we are going to use the lac operon as our example. The lac operon makes the genes, it's going to make the proteins, to break down lactose. Right? So this is only transcribed, translated when lactose is present. When lactose is absent, you don't want to make these genes or make these proteins. So no lactose, and your repressor protein sits on your operator. So, maybe we will go with red there. Oh, come on. So no lactose. The repressor binds to the operator. And so even if RNA polymerase binds, it can't progress because there's a large protein blocking its progress. Again, this is happening on the DNA, on the chromosome. Now, if we add lactose, so if lactose is present, and maybe we will make lactose look like a square. Lactose binds, I should just leave that cap off, the repressor protein. Okay. And when lactose binds that repressor protein, it can no longer bind the operator. So lactose binds the repressor protein. Which no longer binds the operator. So when you don't have lactose, you have no transcription. But when you add lactose, you have transcription. So that's what this cute little image on the overhead is trying to show. No lactose and your repressor protein is bound here at your operator. You add lactose, which is called an inducer. It's going to bind your repressor protein, and it comes off of that operator. So it's really just a physical blockade. So in this case, lactose is our inducer. So that's an inducible operon. Questions on that one? We're just going to use lac as the example. For a repressible operon, we're going to use what's known as the trip operon. And this operon makes the amino acid tryptophan, or it makes the enzymes that carry that out. And so you're going to have the same setup. You're going to have your 3', you're going to have your 5'. You're going to have your repressor protein here. You're going to have your promoter. You're going to have your operator. You're going to have your genes. Actually 5 genes in this one. And you're going to have your terminator. So same setup. You're going to make a repressor protein. I'm going to say we'll make them a triangle again. These operons are normally on. So in this case, if the cell is very low on tryptophan, then the repressor does not bind to the operator. So you have transcription, right? We have transcription. once the cell has made a lot of tryptophan and it's built up in the cell and it's like, hey, I don't need any more of this right now. How do I put use the word present? Tryptophan is going to bind the repressor the same way lactose did over here. But in this case, when tryptophan binds this repressor, that is what binds our operator. Right, so tryptophan binds the repressor. Tryptophan plus your repressor protein bind the operator. So you have no transcription in this case. With the lac operon, lactose was considered an inducer because it induced expression. With the tryptophan operon, tryptophan, that's my little blue box here, is known as a co-repressor because it takes both the repressor protein and tryptophan to make it an actual repressor. And again, that's diagrammed on the overhead. Their drawings are much better than mine. But if nothing else, just think about, do I want it off or on? And if you don't want transcription happening, then you need to figure out how this thing is binding to the operator. I will stop there for a second. Questions on that? That's the easy part. For anabolism, this is pretty much all there is. It doesn't get more complicated. Catabolism gets more complicated. If you were just a single cell and you had a choice of glucose or lactose to break down, which one do you think you would prefer?
Glucose.
Glucose. You're going to get more ATP. So the cell actually has a built-in mechanism to make sure if there's glucose, it will use it instead of lactose. All right. Now, there's some other things. We won't talk about those. All right. So lac operon again. This is just summarizing all this again. Lactose will activate the operon. Okay. So if glucose is present, it will actually quash the transcription of this operon. What I'm going to do, I'm going to expand this out. I'm going to make this a little bit bigger. In a process known as either catabolite repression or glucose repression. All right. We still need our eye to make our repressor. That's no different. Ooh. We still got our repressor protein here. Okay. Oh. You know what? I didn't put it in my overheads. We're not going to talk about how this happens because it's complicated. It'll save you a little trouble. So in that case, I just want you to know if glucose is present, lactose and other sugars will be inhibited. Goodness, those of you who had 161 with me, you had to do this for 161. You don't have to do it for micro. If glucose is present, use of other sugars is inhibited. We'll just leave it at that. All right. Just know that is an endpoint. And that's either catabolite repression or glucose repression. That's what they're trying to show you here. If you're using glucose as a food source, the cells will grow faster than if they use lactose or other sugars as a food source. It's just because you get more ATP per molecule. That's all. Okay. Questions on that? That's a lot easier. All right. That is it for chapter 7. Anything else we can see? Ready for chapter 8? Chapter 8, we're finally going to get to some good stuff, really micro-specific, that will be applicable to disease and antibiotic resistance, much more directly applicable. Hey, come up here. And I will put some ed puzzles up for chapter 8. I thought there was more to chapter 7. So I didn't put much up yet. So what chapter 8 is about how bacteria change their chromosomes. Okay. How do we change our chromosomes from generation to generation? You guys remember your mitosis, meiosis especially. Meiosis, there were 2 sources of rearranging chromosomes when you have offspring. Remember what those were? So prophase 1, did you remember, Sarah? Crossing over of your homologous chromosomes in prophase 1, so mom's and dad's chromosomes get all mixed up. And then it's just sperm and egg coming together, because every single sperm and every single egg are different. And so our offspring are very genetically different from ourselves. Well, how does a bacteria reproduce?
Asexual reproduction?
Asexual. They're all clones. There is no genetic diversification. And that is very, very bad for long-term survival. So this chapter is about how bacteria create genetic diversification. And this is going to be probably completely new information. All right? 2 major ways of creating genetic change in bacteria. One is the same as us, through mutation, actual change in the DNA sequence. And that's what we're going to look at today here in lecture, is what kind of changes mutations can result in. The other way that bacteria can change their genetics is something called horizontal gene transfer. And that would be literally like going into a handshake with another person and giving them some of your DNA. And so literally within a generation, bacteria can exchange DNA. You already learned about one when we talked about viruses. We talked about transduction and how some bacterial DNA can get trapped in that phage head and then get transferred to the next bacteria. That's one form of horizontal gene transfer, but it's probably the least common form. Right. So mutation, just changes in the DNA, and horizontal gene transfer, which is something very unique. All right. First, just basic definitions. Genotype. Your genotype are just your genes, the actual alleles you have. Bacteria have a genotype as well. Remember, they're haploid, so they only have 1 copy of every gene normally, but that is their genotype. And then phenotype is what we see. So if we think about E. coli on McConkey, a fermented lactose, so it has the genes for that fermentation, that's its genotype, and its phenotype is that it fermented lactose. Okay. So we're going to talk about both genotype and phenotype in regards to these changes to bacteria. First type of change are just spontaneous mutations. Tend to happen while DNA is being copied, right? DNA polymerase is going along, adding in new nucleotides. It's not perfect. It makes mistakes. You do not need to know the actual mutation rate here. It differs per organism, but mutations are rare. But when you're copying for E. coli 2 million base pairs every time you go to divide, you know, every few hundred cells is going to have an error in it. If there's an error in that DNA, bacteria, again, haploid, asexual reproduction, every offspring is going to get that error. Right? It will get passed on. Sometimes that same nucleotide will be changed back to its original nucleotide. That's what the word reversion means. We're actually going to talk about reversion in, I don't want to say phenotype, but results today in lab. We'll talk about reversion as well. So reversion is just going back to the original. All right, so spontaneous mutations happen because of errors made by DNA polymerase. And they're rare, but they're not extremely rare. I mean, think about it. How many cells do you think are in a colony when you grow bacteria?
Hundreds?
Actually, millions. There is millions of bacteria in those colonies. If you're starting with one bacteria, and all those times it has to divide to make those millions, there are multiple mutations within that colony. Right? It's not a perfect clone. All right. Let's talk about mutation types. I'm going to get rid of the tripoperon. Yeah, maybe I'll get rid of the lacoperon too. And I know none of our introductory classes here talk about mutations, so I'm going to spend a little longer on this because this is good information not just for microbiology, but just for biology. Our cells make mutations too. It's why as we get so much older that we develop diseases, especially cancer. It's because our DNA polymerase is not perfect either. So the most common type of mutation in the DNA, so DNA mutations, is what is known as a point mutation. Right. Another name for this is a base substitution. And it just means that one nucleotide got replaced with another. The way I like to think about mutations, so right, your codons are a 3-letter code. So I like to just use a sentence of 3-letter words. And the sentence that I have found is the most fun to play with is the cat ate the rat. All right. A point mutation could be something like that. It still makes some sense. It's not the same thing, but it still is a reasonable sentence. It could be more something like this, which makes something that doesn't make as much sense. So point mutations can have no effect. They could have a lot of effect. So let's do a couple options here. And my first one's the hardest one to kind of replace with this, but if we did the C with a K, we put in a new, different nucleotide, but as long as we still pronounce it the same, it says the exact same thing. The term you'll usually see for these is a silent mutation. Your textbook calls them synonymous. And this is what's going to happen with the protein. Protein, E-I-N. A silent or synonymous mutation, so you get a point mutation that's going to cause the same amino acid to be inserted. You know what? There's no change at all to that final protein. You get a nucleotide change, but no change to the protein. And again, your book calls these synonymous. I will frequently call these silent, just because that was the term I learned. I'm going to go back to my cat here. These are somewhat common, but the most common result from a point mutation is what is known as a missense mutation. And a missense is where you get a different amino acid. So that would be where I actually change what it means. So a different amino acid is inserted. So you change a nucleotide, you're going to get a different amino acid. This is the most common result of a point mutation. Missense mutations are usually detrimental. Usually the protein doesn't function or it's less functional. So usually detrimental. And most of our genetic diseases, or many of them, are missense mutations, where the protein loses its function. Sometimes they have no effect on the function. If you're changing one amino acid with another one that's a similar size and charge, it's probably going to behave similarly. So there might be no effect. Or in rare cases, there's actually a gain of function. The protein becomes better at what it does or even gains a new function. Usually detrimental, but we'll get some other things happening. All right. So if at the end of my sentence I had a stop codon here, and if that got changed, now it's no longer a stop. That's what we call a nonsense mutation. And that can work the other way as well. If, I can't really do this very well here, but if the stop codon got added early as well. So adding or removing stop codons. That's your nonsense mutation. If you add one, the protein just got shorter. It usually loses its function unless you only lose a couple of amino acids at the end. If you lose your stop codon and this thing goes on, you also usually lose function. So these almost always lose function. nonsense codons are the worst in terms of effect on the cell. Okay.
These are all codons?
Say that.
Just the nonsense one?
So nonsense is just if you add or remove a stop codon.
Oh, okay.
So anytime the stop codon gets messed up. And that's almost always very bad. All right. Questions on those? So those are all possible results of just changing a nucleotide. There is a worse type of mutation out there called a frame shift. So if you add or lose a nucleotide, addition or deletion of nucleotides, I would say of a nucleotide. Well, if I take my sentence and I lose this T in cat, now I've got the CAATETHEB, whoops, AT. You get the idea. Everything past that addition or deletion makes no sense. In the protein, I'm sorry, not in the protein, this is what we call a frame shift, because the codon reading frame has changed. Frame shifts almost always result in nonsense. Stop codons usually get lost, and you get these gigantic proteins. Did I want to say it? So the best example, that was what I was going to get to, now I remember. If you think about a DNA sequence, you remember thymine dimers? When we hit the bacteria with the UV light, and it killed the bacteria mostly because they made thymine dimers. So anytime you have two thymines right next to each other in the presence of UV light, they actually covalently attach to each other. And when the DNA goes to be copied, it will only see that as one nucleotide. And DNA polymerase, for whatever crazy reason, has a behavior of putting in adenines if it doesn't know what else to put in. So here, instead of AATT, you would get AAA. And then it would jump to CGC. So this is why UV light leads to skin cancer so much. It's because it causes thymine dimers in our skin. And then you get frame shifts. That is the main source of those, is thymine dimers from UV light. See if I had that in there. I have it in here just a little bit further. So know the 4 types of two types of DNA mutations, 3 results here for proteins. Said these are spontaneous. You can also induce them, right? Anything that's a carcinogen induces mutations. That's what a carcinogen is. We're not going to do a lot with this. You don't need to know the details, but chemical mutagens would be any chemical that induces mutations. And I've got some examples in there. I'm not going to ask you specific examples. All right. But you can make guanine look like something slightly different by adding a methyl group, and then it'll pair incorrectly. And I know adenine, you can do that as well. So chemical mutagens just change the nucleotides enough that DNA polymerase makes mistakes. There are also, again, environmental mutagens. Oh, there we go. Like the UV light making thymine dimers. And again, DNA polymerase is going to make a mistake there. All right. Give you a few examples. I'll jump back. So, some chemicals look like nucleotides, and DNA polymerase will add them, and they pair incorrectly. So the only real difference here is these chemical mutagens change the nucleotides that exist within your DNA. An analog is a chemical you introduce that will replace the nucleotides in your DNA. These are all carcinogens. The worst carcinogens are these things called intercalating agents. So if you were in 161 and we ran the gel, I had you guys wear gloves because I didn't want you touching the stuff that stains the DNA, because these molecules slip between base pairs. And then when your polymerase goes to copy, again, it often sees these as nucleotides and will add nucleotides to your DNA. So intercalating agents tend to cause frame shifts, whereas those base analogs and chemical mutagens tend to cause point mutations. So these guys are the worst mutagens. We don't have a lot of those around anymore. We've tried to get rid of them.
Say that last part again.
So base analogs and chemical mutagens are going to create point mutations.
Yeah, in the last part.
And then these intercalating agents are going to cause frame shift mutations.
Thank you.
You're welcome. I'll come back to these guys. The environmental mutagens are radiation. Talked about UV light, thymine dimers. X-rays, literally, like if you go get an X-ray, it breaks DNA. It literally breaks the sugar phosphate backbone. So I usually wear a big lead vest when you're doing an X-ray, except for the area they want to X-ray. Right. And breaking that backbone is generally deadly to the cell, or the DNA or the cell can't repair that. That's why X-rays are great sterilizing agents. One other thing, and I'm just going to introduce these. I put them a little bit in your smart books and your assignments, but not too much. Are pieces of DNA called transposons. The simple name for a transposon is a jumping gene. Transposons can have a gene within them, but there's a segment of DNA that can literally pull itself out of the chromosome and move to another area of the chromosome and put itself in. Some transposons will actually make copies of themselves and jump around. There are transposons within our own DNA. We've found these in almost every living thing. Two uses for these: they can cause major mutations if they insert in the middle of a gene, right? It's going to inactivate that gene if it does that. But again, if your transposon is carrying a gene, like antibiotic resistance, it can be used to pass that gene from organism to organism. We're not going to say a lot about transposons. Just know that they are another source of mutation. If you ever want to read good biology history, the discovery of transposons was another very interesting part of biology history. Questions? So you have mutations can be spontaneous. They can happen through a variety of chemical mutagens, environmental mutagens, transposons. These are the results. Usually bad. That's kind of the take-home message. Cells can repair some damage, and we're not going to do a lot with this again. All right? So some damage can be repaired. First, DNA Pol 3. He's going along, he's adding nucleotides. Why does A pair with T and G pair with C? Yeah, why is it always ATGC? What if you have AG? What happens?
They can't hydrogen bond, right?
And if they try, what do you think happens to this backbone? Yeah, it'll either have dips in or bubbles out, right? You don't have a nice, pretty double helix. So you might have something that looks like this. Well, you know what? DNA polymerase is going along and it's like, that ain't right. DNA Pol 3 can actually, right when it adds this wrong nucleotide, take it back out again. It has that proofreading ability. And it can fix that mistake. Now, if it doesn't, if it keeps going, and it puts a G in there and you get the bubble and then it's all fine again, all right, DNA Pol 3 can't do anything at that point. Once it's added more nucleotides, it's stuck. There's another DNA polymerase called DNA polymerase 2 that goes along later and looks for these weird bubbles within the backbone. Right? What we call proofreading. Right. So mismatch repair is what Pol 2 is going to do. So Pol 2 is going to go along and it's going to see this weird bubble. Well, it has a problem. How does it know which strand is correct? So you might not know this, but your DNA, the old DNA you have, actually has methyl groups on it. Sometimes they silence genes, if you learned about epigenetics at all. But DNA Pol 2 can use that to say, oh, that's the old strand. I want to use that as the correct template. And it's going to come back and it's going to cut out everything in this chunk, usually about 10 nucleotides, and just replace them all. So that is one nice bit of repair we have. All right. That's mismatch repair. A similar thing is base excision repair. Don't worry about the difference. But they're really the same thing. Right. Thymine dimers. There's actually a specific enzyme within most cells to deal with that, called photolyase. So photolyase uses light as an energy source, and photolyase will actually break these covalent bonds. So you're actually much less likely to get skin cancer from direct sunlight, where you've got white light, versus going to like a tanning bed where you're getting direct UV light and you're not getting the white light to activate this. That's photoreactivation. If that doesn't work, cells can do another thing very similar to mismatch repair called nucleotide excision repair. See, they're all pretty much the same. You cut out the chunk, you replace it with good stuff. Your problem really comes if these are both old strands and you get hit by a chemical mutagen that maybe makes this G look like a T. Now DNA polymerase doesn't know what's old and what's new, and it has to guess. And it will guess. And so it has a 50% chance. The last one I want you to know about is this SOS repair. SOS repair happens when the damage is huge. Especially X-ray damage where you break the backbone and like the cell's trying to put things back together, or a lot of X-ray damage where you've damaged a lot of the nucleotides in a row. Literally, it will just cut out a huge chunk of DNA and start guessing. And this almost always results in major problems, and usually results in cell death or cancer. And that one, there's a lot of enzymes involved. It's not just DNA polymerase going in there and cleaning it up. I'm trying to simplify these. So again, cells do have a way to deal with this, but the longer the damage stays, the harder it is to fix. I'll stop there, because it's like 52 according to that clock. We're gonna go to lab, we'll get everything started for